Hello. Today I will be talking about how knowledge of microbial diversity, in particular seed and soil microbiomes, can be used to inform restoration practices. I will present a brief overview of the field and highlight gaps in knowledge that I will be addressing throughout my PhD. In restoring ecosystems, we aim to create functioning, diverse, and resilient ones, which have the key ecosystem attributes present. In order to define and understand what these key attributes are, we would often use a reference ecosystem. Comparing the diversity of the restored site to that of a reference site provides a way of assessing or monitoring whether our restored site is reaching the benchmark endpoint or at least is on the right trajectory to do so. But what key ecosystem attributes need to be present for an ecosystem to be a functioning and diverse one? And what attributes should we use to indicate whether an ecosystem is on track? Maybe the number of plant species present, the percent cover of different vegetation strata, or the diversity of birds, mammals, or insects present. All of these are often used as indicators of whether restored ecosystems over time have become increasingly similar to their reference ecosystem. But there is one key ecosystem attribute that is less commonly used or even considered in restoration. That is microbial diversity. Microbial diversity is comprised of free-living microbes that exist in the soil and air and host-associated microbes that exist within and on plant tissues, in particular endophytes, which are microbes that live within the living tissue of the host plant, are abundant within ecosystems worldwide and have been found in most plant species studied to date. This microbial diversity contributes to ecosystem function and productivity and forms the microbiome of our plants. The microbiome, as is shown here, is divided into the below ground and the above ground components, with each component consisting of multiple spheres. The plant and soil microbiome is associated with improving plant growth and increasing stress tolerance, improving the resilience of the host and the overall ecosystem. Increasingly, microbial diversity is being incorporated into restoration, with a particular focus on soil and root microbial communities. These communities have been integrated into restoration programs in one or two ways. They've been used as seed or soil inoculants using a variety of methods, such as the transplanting of topsoil from a site prior to restoration or even from a local reference site. They are also incorporated through direct inoculation with often mycorrhizal fungi, and this is done via sprays or dredging. Seed priming techniques, where the seed itself is coated with a single or multiple beneficial microbial species, is a current area of exploration also. Soil and root microbiomes are used in restoration assessment. One commonly used approach is where the dynamics or compositional changes of the soil or rhizosphere communities are tracked along a chronosequence of restored sites representing different ages. These sites are then compared to a reference site, which is often representative of a healthy and resilient ecosystem. The trajectory of these communities along the chronosequence has shown varied results in Australian ecosystems, with these results often being ecosystem dependent and influenced by factors such as the soil chemistry and the plant community that's present. These studies give us an insight into the below-ground microbial diversity of ecosystems. But as you can see here, the plant microbiome also extends into the above-ground plant compartments, and this is termed the phyllosphere, as the stem, leaves, flowers, seed, and fruit of a plant also contain a microbial community. Seed in particular is important for restoration. It is used for broadcast seeding for large-scale restoration projects, particularly in grassland ecosystems. And the growing need for seed in restoration is evidenced by the growing number of seed production areas. Seed is also the basis of many ex situ collections for most Australian native species. So understanding what microbial diversity is present within seeds of native Australian hosts may play an important role in their conservation and restoration projects that utilise these species. Seed microbes, such as endophytes, occur within all the different compartments of the seed, with some studies suggesting that each compartment may hold a unique community. Endophytes within the seed of a plant have been associated with breaking seed dormancy, improving rates of germination, and playing an important role in root development. But they've also been associated with existing as latent pathogens and saprotrophs. In my honours research, seed from 2 Banksia species, indicated by the column graph here, from 4 geographically distinct sites, showed that a core seed microbiome, which I defined as a series of highly abundant species, was conserved from site to site, with this core unique to each of the Banksia species. Currently, we have no reference ecosystems for the endophyte diversity for seed of natural ecosystems of native Australian hosts. For my PhD, I aim to survey the seed microbiomes from multiple populations of Australian native hosts along the east coast of New South Wales. This will give us a baseline understanding of reference ecosystem diversity for seed microbiomes and whether different populations of a host house distinct microbial communities. Through repeating this sampling at restored ecosystems, I aim to see what seed microbial diversity is captured in our restored sites using different ages and different site conditions. In particular, I'm really interested if a core microbiome is present in natural sites and whether this core is then captured in our restored sites. Understanding the functional diversity of the microbial communities in seed may also give us some insight as to how these communities may contribute to the growth and establishment of their hosts and the functioning and productivity of our ecosystems. Seed endophytes have been shown to produce auxins and gibberellins that can promote germination and help with breaking seed dormancy. They can produce siderophores, which play a role in the bioavailability of iron to plants, and they have the ability to solubilize phosphates. Lastly, seed endophytes possess enzymes such as cellulases, amylases, and xylinases that allow for the breakdown of cellulose and other plant materials, contributing to carbon cycling within the ecosystem. I plan to undertake trait-based phenotype assays that are shown here to see whether seed endophytes of native Australian hosts also possess these traits and whether these traits differ between natural and restored ecosystem hosts. It is unknown how restoration practices may affect what microbiome is present in seedlings. The initial plant microbiome is acquired during the germination and establishment phases of a plant, with microbial sources coming from the soil, root, and seed microbiomes. The space around a germinating seed, called the spermosphere, is a dynamic space comprised of interactions between these microbes and with a range of signaling compounds released from the plant, especially during germination. These interactions in the spermosphere play a crucial role in forming the initial microbiome. Little is known about the relative contribution of the seed and soil microbes for native Australian hosts. I am interested in understanding how restoration choices, like seed and soil provenance, affect the resultant seedling microbiome and overall microbial diversity of a site. I aim to investigate this using a series of greenhouse experiments where I manipulate where the seed and soil are sourced from, grow the seedlings to a couple of months old, and identify using NGS techniques what microbiome is acquired by the plant as a result of these manipulations. I then want to see if this is correlated with any differences in plant growth. Firstly, I'm interested in understanding if removing the microbes from the seed and soil has any effect on the formation of the seedling microbiome and whether this correlates with differences in germination rates, root growth architecture, and seedling growth rates. Secondly, as using locally collected seed is one method of selecting what seed to collect to revegetate a site, I aim to better understand the role that seed provenance plays in restoring the seedling microbiome. Does collecting seed and soil from the same site result in a microbiome more similar to that of a natural ecosystem reference host? And is the resultant microbiome correlated with improved seedling growth? What is the resultant microbiome if non-local seeds are used, or if seed from a nursery is used? And does this have any effect on plant growth? Studies where microbes have been added to the soil to improve germination rates and seedling growth have shown that the resident soil community can be more beneficial than adding non-local microbes, with some studies reporting only improved growth when using a locally adapted microbial species. So having some idea of the local microbial provenance of a host may inform seed collection guidelines and ex situ seed conservation practices. Apart from gaining a better understanding of microbial diversity of native Australian hosts, in my project, I would like to work towards providing answers to the following questions, directly applicable for seed collecting for restoration and ex situ conservation. So, if populations house distinct microbial diversity, how can seed collections capture this? Are both seed and soil microbes equally important to forming the seedling microbiome? Is collecting local seed best for the seedling microbiome? And do we see improved growth rates? And lastly, do wild collected seeds rewild the microbiome better than nursery seed? As I am only a couple of months into my PhD, I'm interested in your feedback and suggestions, and I hope I've provided some insight into why conserving and restoring microbial diversity, in particular seed microbial diversity, may play an important role in creating functioning and resilient ecosystems. Thank you.